Final speaker of the session is Ann Willis.
Thanks very much for inviting me to give a talk today. I'm aware that people are generally interested on how you might profile the translotome in various tumour samples, so I'm going to give some examples of what we've done in our lab and what we've learned and what sort of things you can look for and how to take these things forward in general. So as been said, this is protein synthesis, you've got 3 stages: initiation, elongation, termination. Every step is really highly regulated, and from all the tumours that have been looked at, is a lot of evidence that overexpression of the factors that are required for these processes drives tumorigenesis itself. So majority of cancers, protein synthesis is dysregulated. It's not looked as much at as transcriptional control, but this is just a general phenomenon that happens. So therefore, if you target mRNA translation, it's going to be a great way of creating some new therapeutic targets. And I'd actually argue that if you, sort of from what was following on from what Nahan said, that actually if you block translation, initiation or elongation, you're downstream of all those signaling pathways. So one, it's actually more sort of pan-wide way of turning off protein synthesis and controlling tumour growth, but also you have much less chance. These things are not redundant. You feed in from multiple signaling pathways, but you're hitting something right at the bottom. So there's less chance to get resistance and escape as well if you hit these pathways. And also it's going to be cancer cell specific because these are really highly upregulated in all tumours, therefore it's a way of dampening down. You've got quite a good therapeutic window if you start targeting initiation, elongation factors if they're hugely overexpressed in tumours. So we're quite interested in looking how you can look sort of generically at what's dysregulated or not. As I said at a talk yesterday, when you look at transcriptional changes, you're looking at about 50% of things actually validate, the rest don't. And that's because you get a huge amount of translational control goes on in normal cells, but also in tumours. This is just to kind of show you. So there's 2 ways of doing this, and they're both alluded to in the previous talks. So one is polysome profiling. This is the easy way of doing it. And it just depends on what information you want to get out as to which method you choose. So on this method, you take an lysate, you spin it on a gradient, and things that have lots of ribosomes associated with them are very highly translated, generally speaking, and things that very few are not. So therefore, if you fractionate through the gradient, this is what you see. So this is a 40, 60 ATS ribosome. This is a double subunit here. And this is when you have 2, 3, 4, 5, 6, etc., polysomes associated. So this stuff here is very highly translated, and this stuff here is not. So either you can take this and put it on a chip in old-fashioned experiments, or else you can put it on, you do RefSec of the pools. And so what you get, if you look at a ratio from the subpolysomal region to the polysomal region, is a ratio of what's being translated, which is independent of transcriptional changes, because often things that are up at the level of translation can also be up at the level of transcription as well, and you don't want that to confound the data that you get through. The other way of doing it is ribosome profiling. Now this is great, as Jamie showed you. If you're really interested in stall sites or things that might be controlling elongation events, so you're looking for pauses, then you really have to do ribosome profiling. Otherwise, I probably wouldn't, because this takes a lot longer to set up, and also the bioinformatics associated with this is much more complex. So if you just want to ask a neuroblastoma what is and what isn't translated, what does the dysregulated translator look like, I'd actually go for polysome profiling. It's an easier technique to do if you're not expert in these sorts of things. So for ribosome profiling, again, you isolate your ribosomes, you treat with an RNAs, and then you treat them with a protease. This is what you see here, it's a typical sort of trace. So this is our profile here. You can purify this stuff, which is the polysomes, the things that are well translated, and then you treat with an RNAs, and therefore that will isolate your the ATS ribosome subunits with protective fragments. You can run it on gel again. You can then take this peak here, and then you treat the protease, and you end up with ribosome protective fragments. And then you make, which is on here, then you make a library, and you sequence, and you compare those sequences back to your reference sequence. And so this is a typical data that you have. So what you should get, if this has worked properly, you should see good periodicity, which is what you can see on here. So as you get the ribosome, you get periodicity as the ribosome reads across the individual codons. And of course, the way we originally did this experiment was we actually added cyclohexamide to stall things, so you get things protecting the fragment. The one disadvantage of that, you get a very, very large peak at the AUG. That's been found by many groups. Probably now it's better to just freeze your samples to actually store them in a more reproducible way or more biologically relevant way. And of course, you shouldn't have reads down in the 3' UTR, and you may get different interesting reads in the 5' UTR, depending on where, if you have upstream AUGs, open reading frames on there. Okay. So it provides very, very robust data, but if you want to do this in neuroblastoma, this is what you need to do. Really, we've done this from quite a lot of tumor types. We got the best data when we've used at least 6 biological repeats. So take 6 tumor samples and use those. And if you do technical repeats from those, better still, but you want a lot of samples to get this right. Don't take random tumor cell lines and think this is going to work, because it won't. You'll have things that are far too different, and it'll be really, really hard to work out what's going on. So actually, ideally, if you can get your samples that are maybe transcriptionally similar, or for example, in one thing I'm going to show you, when we looked at diffuse large B-cell lymphoma, we took lymphomas which had the same translocation, and that worked really, really well in terms of actually working out what was going on and getting a robust data set. And where possible, I really would suggest you can use primary tumor lines. We found, just from a couple of early experiments we've done, where we've used commercially lines, they've drifted so far from the translatome and the transcriptome from actually the tumor. And if you want, it's fine if that's what you want to ask, but if you really want to look at a tumor sample, if possible, make some primary lines yourself or get some dissected material from actually patient samples themselves, and that would probably give you the best data. So we've done various ones. I've just picked 3 things that we've done because I think these 3 emphasize the sort of differences that you can see in actually the translatome in different tumors. So I'm going to talk about, we have examples of dysregulation, elongation in the similar way that Paul has, but just for this talk, I'm going to give examples where we're looking at dysregulation of initiation. So as Nahum said, you need initiation factors, you need the pre-initiation complex, and overall this is really controlled by phosphorylation. Within the complex itself, you have 4E, which is a cap binding protein that Nahum talked about. You have a dead box helicase 4A that's required to unwind secondary structures. Then a whole bunch of other accessory proteins that are required to regulate this process. So, for example, poly A binds a poly A tail, 4B binds 4A and PAB and helps unwind the structure and stabilize the complex. I'm going to show you about that in a minute. And EF3 binds 4G and the subunits. You need a whole bunch of things to come together. Don't worry about what they do, but I'll just sort of show you a little bit in a minute. And as Nahum said, very, very often in cancers, you have dysregulated signaling via TOR. So all these pathways converge on this. And that's why, actually, if you want to target something in a tumor, you're far better seeing whether you can hit these rather than up here, because there's so many things feeding in, you can get resistance pretty quickly. And in particular, there was a lot of hope with some of the rapalogs that were being developed. And actually, clinically, they've not done very well. Partly it's because there's been resistance and so many other pathways feed in, you can override some of these things. But also it's possible as well that there's not been enough understanding of the tumor biology, and they've not been actually used in an appropriate way. Probably if there's better patient stratification, you might have got better usage of some of these compounds. Okay, so this is diffuse large B-cell lymphoma. What we did, this was cell line based, but we took 6 cell lines that contain the same translocation, and we actually did 3 technical repeats, and this is polysome profiling, so we did 18 samples. We got a really, really good data set from this. And what's important, what we always do, we go back to tumor samples to make sure what we see, if we use a cell line model, is actually also true in patient material. And we published this quite a while ago now, but it's still quite a nice study. Now, this is quite simple in a way, because the driver for this tumor in terms of translation was EIF4B. So this is a protein that stimulates 4A to unwind secondary structure. It also binds poly A binding protein and actually stabilizes the whole initiation complex. We also showed that it's got a terminal oligodenine polymerase track in its 5' UTR, and it's actually downstream of TOR signaling. So we kind of have like a feed forward in this tumor. So we have upregulation of TOR, which stimulates translation, which then stimulates more translation. And that seems to be quite the case in a number of tumors that we look at. You get sort of feed forward stimulation via dysregulation of the translation machinery. So one thing that you can do if you're interested in neuroblastoma is just take some cell lines, or actually better still tumour samples, and just ask the question, is there increased expression of initiation factors, and if so, which ones? And you will see them for sure. But in this case, we had, surprisingly, not anything else, not 4E, except for not 4A, but we had an increased expression of 4B in our cell lines. We also had increased expression of 4B in our tumour samples. And what we found was going across from grey, so going across from follicular lymphoma to late-stage follicular lymphoma, which then can develop into diffuse large B-cell lymphoma, we gradually see an increase in 4B expression. What's it driving? We actually find that it drives translation. We were looking at kind of key groups of things and things that might be associated with resistance to therapies. And we find that actually we drive expression of proteins that will protect from apoptosis. So, for example, we've got high expression of BCL2, you can see here. It actually drives MYC expression. So that if you look, these are the RNA. There's no change in the RNA, but we've got enormous increase in expression of the proteins. We also find we get an increased expression of enzymes, the DNA repair enzymes, that actually would drive resistance to various therapies. So, for example, ERCC5 is hugely overexpressed in the cell line model, but again, in the absence of any change in the RNA. So the RNA is the same, the proteins are up because you're driving the translatome, not the transcriptome, by having high 4B. And again, we've managed to validate what we had either on tissue microarrays, or these are just samples from patients, from a biobank. And we have high BCL2, high DAX, high ERCC5, for example, in our tumour samples. What's important is actually there's a correlation between autoexpression of these things and survival of patients. So overexpression of ERCC5, DAX, and 4B is associated with poor prognosis. But particularly patients who do very badly initially tend to have high levels of expression of these things. It's sort of driving a pretty aggressive form of the disease. So what we actually showed as a pathway is in what seems to be triggering this whole cycle is actually activation of TOR, which increases 4B expression, and then that increases expression of these proteins. And what we think is happening is it's not specific sequences in here that's doing it. We think what it is actually having something that stimulates 4A function is actually increasing the width of the translatome. So all these things have got very, very highly structured 5' UTRs, as do lots of proto-oncogenes and etc. And actually what you do is you just allow their expression to go up by just having that much more of the things that would be allowed to unwind them. And I think that's, whilst 4A seems to be binding to, ERF4A seems to bind to some quite specific sequences, we don't think that we're seeing this case with 4B. And that itself appears to be associated with drug resistance, which is why we think those patients who have these high level things do so badly within the first year following diagnosis. This is a very different sample. So we looked also in peripheral blood chloric lymphocytic leukemia. And these cells are very weird. So actually in the blood, they're pretty quiescent in comparison to when they get stimulated in the bone marrow. And this time we took peripheral blood and we purified the CLL cells from 42 patient samples. We didn't do technical repeats because we had so many patients we used. And then we used a separate set of patient samples to actually validate the data that we had. Okay. What we found in here was actually we weren't surprised because the cells are quite slow growing. So compared to our normal purified B cells, we tend to have a downregulation of translation. But in particular, we have a downregulation of various selectively of ribosomal proteins, which we're very interested by this. So the ribosome, as was shown on the nice pictures from Jamie, it's a very complex molecular machine. It's got a large and small subunit, and there are over 80 different ribosomal proteins. But what's become quite popular and very exciting in the area of translation is actually not all ribosomes are the same. So originally, we just thought they're these molecular machines that are going on and do their job. But actually, it seemed that different ribosomes have different components of RNA binding proteins associated with them, and they have differential functions. So some lovely work suggested, if you're interested, to have a look from Maria Barner, who's actually shown what some of these different ribosomal proteins do and how they drive different expression patterns. And also, it's known that if you have deregulation of dysregulated expression, of individual subunits of RNA binding proteins or ribosomal proteins is associated with tumorigenesis. So RPL19, for example, is associated with various types of leukemias. So there's been data suggesting that actually these slightly weird ribosomes that don't have the full complement of ribosomal binding proteins are sort of cancerous, and somehow they allow selective translation of subsets of messages. So what we found is we've got downregulation, initiation, and ribosomal factors, but not all of them. It's subsets, which looks like one of these sort of ribosomopathies where you have altered things. What was really odd, and we don't know what this means, that in all of these other, so the kind of purpley is the CLL samples, we actually have this weird higher form of ER4A, and we have no explanation for this. It's the case in nearly all the CLL samples we've looked at. We do see some downregulation of 4E and 2-alpha. It's more dramatic for some of the ribosomal proteins that we have. So, for example, RPL9, we've got a decreased expression of that. If we take this all together, we've done stats on all this data. So these are the ones that we find seem to be selectively downregulated in our leukemic cells from patients with CLL. So this is the 4B, 4E, EF2A, RPL7A, etc. And then we also found we have a decreased expression of discarin, and we were quite interested in this because this is a protein that's required to actually modify the ribosomes. And again, altered expression, this is associated with a particular sort of cancers. Oops. So not surprising, we actually have, because we seem to have a sort of overall downregulation of ribosomes, we've got a decreased number of nucleoli in our cells, and we actually showed that we actually have seems to be a defect in ribosome biogenesis per se, and so we're seeing a difference in the maturation of the 21A ribosomal RNA. It was impossible, we couldn't, because we're working on primary cells, we couldn't then sort of knock down or overexpress DKC1 itself. It's just too hard to do. So actually we took cells from patients with dyskeratosis who have low levels of DKC1, and this is what these are here. And actually in these patient samples, we also find that we have, weirdly, this slightly higher band of 4A, but also we see downregulation of the proteins that we see in our CLL samples, so RPL15, RPS8, for example. So what we think we have is actually, it could be that it's DKC1 that's somehow modifying this whole program and is one of the drivers. And then if we look at survival samples, we find that patients who have high levels of discarin, so towards more normal level, have a much better overall survival following treatment than those that have low level of this protein. Okay. And then, so this is our last example. So this is malignant pleural mesothelioma. One of the reasons that we are interested in working on this is that the UK was uniquely exposed to asbestos. It was very much embraced as a building material, and we have probably the highest rate of malignant mesothelioma in the Western world, which follows exposure to asbestos. It's a very, very, very odd disease because it's got a 40-year latency period. So people who are exposed in their 20s don't develop the disease till they're 60. But once it develops, it's very, very rapid. There's absolutely no treatment, and people generally die within the year. So in my unit, we're interested in this. We've made a lot of primary cell lines from this to actually start understanding and start unpicking what's happening in the disease. We've also got animal models where we're looking at early stage, but these are just all from patients with end-stage disease. So we've made about 20 primary cell lines, and in this experiment we used 6 that were transcriptionally similar. I'll show you in a minute. It's a very rapidly evolutionary disease as you go through, even growing in culture, and we did 3 technical repeats of this. So this is what I mean. So these are the primary lines. This is some of the transcriptional profiling data. So we took the ones that were more transcriptionally similar. This is what I was saying a little bit about, just be very careful about using commercial lines. So when you look, these are our primary lines. This is principal component analysis. They kind of roughly clump together. These are the immortal lines, so they actually bear no transcriptional similarity at all from the primary lines that we've made. They're just too far and too many changes happening in culture. That's not going to give us any useful information about primary disease. And this is our primary untransformed mesothelial control samples here. So we did the profiling on these. We also looked at what was up and expressed. We have certainly activation of TOR going on in this pathway. We've got a very, very high level of methionine incorporation. These cells are growing really, really fast compared to our normal mesothelioma cells, which, it's an outer layer. They barely grow at all, the primary cell samples. We have some different things, again, that we weren't expecting to find that we were translationally upregulated in these samples. Again, we have translation is up. As I was saying, it's a feed forward. So very often you have translation up, and it's driving the translation in the tumours. So you've got translation up, and we've got ribosomal proteins are up. That's also very common in a lot of cancers. We're very surprised. One of the things we had up was mitochondria and respiratory electron transport chain. We'd not seen that in other tumour samples that we looked at before. So we decided we'd investigate that. I think, here we are. So this is just proving that it works. We've got high levels of ribosomal proteins. You've got to have more ribosomes to double. So tumour cells often dysregulate ribosome biogenesis. This is being really driven forward. It's all up very high. What was compared to the first example I showed was a diffuse large B-cell lymphoma, where we just seem to have 4B that's high, and that seems to drive quite a nice tidy story. We seem to have a lot of the initiation factors up in malignant mesothelioma. So we've got high levels of 4A, we've got high levels of 4G2, we've got high levels of ERF6. So we've got some things seem to be very, very up, and it's been driven almost from lots of points along the initiation pathway. When we looked at what mRNAs encoding respiratory electron transport chain were up, we found a lot. So, tons. It was just universal right the way across. There's something really odd going on with the mitochondria in here. But when we looked at, so this is just showing that those proteins are up, there's no change in the RNA. And when we looked at the mitochondria, there's something very odd about them. So they're really, really enormous compared to normal mitochondria. So this is a normal one here with the red dots. They're all absolutely huge. But in fact, there's not more of them. They're just much bigger. So there's some really big, so having this sort of increased expression of these proteins seems to have driven this some really, really big fusion event that's going on. But you can do various seahorse experiments, but one thing that is kind of quite tidy, one thing you can actually do is if you can force cells to grow on galactose rather than glucose, you make them use their mitochondria. So when we did that, we were very surprised that because we got these really big mitochondria, we thought they might be better, but they're not. You can't sustain growth of these on galactose, where other tumour lines, this is a lung tumour and HeLa cells, after a little while they do adapt to growth on galactose. These don't, they die. So whatever it is about those very big mitochondria, they're not actually functional. And then you can actually start combining things together and using stuff to see whether or not you can actually use this as a way of selectively inhibiting tumour cell growth. So, as I said, these cells are really resistant, and this is well known. There's very, very high doses of cisplatin here. But when you start inhibiting, not with Patamin A, but with hyperistenol, we can actually start decreasing cell survival. So you can start doing combined targeting of translation initiation along with some other classic cytotoxic drugs and start to get some cell killing. And this is one of the first times we've actually seen any cell death at all in our tumour cells. A slightly better way of doing this, and we've worked this up quite well in our unit, is what you can do is you can take explants from patients. So you maintain the 3D tissue architecture. And I really recommend if anybody wants to try treating samples, actually see what they're getting. This is a really good way of doing it. So what we do is take explants, and then we treat, and you can maintain these for 4 to 5 days in culture. So this is untreated. So we added taurine, so we're inhibiting signaling through TOR, or we added galactose. And this is Q67 staining, so we're just looking for cell growth. So you find when we add these, we actually start slowing down tumour growth. When we add taurine and galactose, so we're inhibiting translation and we're also inhibiting these weird mitochondria, we actually end up with static. So we inhibit tumour cell growth completely by adding both these things together, which may be a way forward of providing some treatment option for these patients who don't have anything else that you could treat them with. This might be something we could do. So this is kind of very complicated, this one, compared to the other slightly cleaner ones we've looked at. There seems to be multiple signals coming in to stimulate translation in these cells. We certainly got TOR is up. We have high levels of 4A. We've got high levels of ERF6. What's quite interesting is Stefano Biffo did the kind of reverse experiment when they knocked out EF6 in a kind of muscle model. They found they actually affected metabolic enzymes, and they end up with very, very odd mitochondria. So it's almost as though we've done the reverse. It's a sort of like a reverse situation in this tumor. Okay, so generally speaking, these are the types of things you see dysregulated in your tumors. If you look, I would look at a range of initiation factors, but you will find that they will be up. Also, it's worth looking at ribosome biogenesis factors because, again, that's very, very driven in aggressively growing tumors. It actually makes the cells, because you increase the width of the translatome, and in lots of cases you increase expression of anti-apoptotic proteins like the BCL families, it often makes them very resistant to apoptosis. But you can, with this understanding, you can go in and start trying some new ways of doing dual targeting to get either chemostasis and no growth, or actually tumor cell killing. Okay, so protein synthesis is dysregulated in all cancers. It's very disease specific. Very often, increased signaling through TOR, because so many metabolic signals that's required by the cancer cell come from that point, drives a dysregulation. And what we've often seen, and we have a feed forward, so whilst we look at the initiation factors, the first off, often the initiation factors are showing increased translation as well, so you get more and more of them produced. If you do ribosome polysome profiling, if you just want to look at what you have is up in the translatome, I really recommend you do polysome profiling. If you want to look at message-specific pausing, then you need to do ribosome profiling. You require good biological repeats, so don't start off just taking commercially available lines and profiling those, because you'll just get messy data out and it's not worth it. It's better either making your own primary lines or doing some microdissection from tumor samples and using those instead, and getting material that's at least transcriptally similar gives yourself a fighting chance of actually getting some important things back. But also, from some of the other work we're doing, targeting protein synthesis is going to provide some new really exciting therapeutic avenues. And as Nahum said, if you knock out 4E, you stop everything. You don't have to worry about what's feeding in as an upstream target, or 4E phosphorylation, etc. So this is going to be a new way forward. There's lots of drug companies now are really, really interested in the translational machinery, and I think this is something that could be very applicable to neuroblastomas. Just remains for me to thank the people who've done the work from my lab, and we work a lot with Owen Sampson on various mouse tumour models. Graham Packham from Southampton has been really helpful with diffuse large B-cell lymphoma samples. And John LeCain is our unit pathologist who helps us get all the patient samples, make sure that what we do is actually translatable and is human relevant. And thanks for listening.
Thanks, Anne. I just wanted to ask you about those mesotheliomas with those big mitochondria. The obvious question is whether they're really glycolytic, or, if they're not using glucose or nutrients for oxphos, as you showed, and if you force them to use galactose, what are they using normally?
Well, I think they're probably using glucose in their patients themselves. They're just not using. You can't use other source of energy.
So that's what I mean. They're probably glycolytic then.
Yes, that's right. Yeah, very high.
Right, or using fatty acids, for example.
Yes.
Or,
other stuff's feeding through into those.
Okay, thanks.
So in the mesothelioma, do you see a difference between 4A1 and 4A2 because they have two functions? Because you measured both, right?
No, it's just 4A1 is up. We don't see a change in we don't see any change in 4A2. It's just. So
you don't see a difference in 4A2?
No, just 4A1. It's EF 4G2 is up, not EF 4G1. Which was unexpected.
Okay. Thank you.
I'd like to thank all the speakers for a great session and all the audience for staying.